Hey guys, today we're going to learn about passive transport. One of the ways that your cell membrane is able to transport or move molecules into or out of the cell. Let's do this. What is passive transport? Passive transport is defined as the transport of movement of molecules across the cell membrane without the use of energy. Remember, the cell membrane's job is to control what comes in and out. There are going to be molecules within the environment that the cell is placed in the extracellular environment, as well as inside of the cell that the membrane will have to regulate the direction it moves. Passive transport will allow the cell membrane to permit molecules to enter or leave depending upon the amount of molecules located either inside or outside of the cell. And because it's all based upon the amount of molecule inside or outside of the cell, the movement of those molecules can take place without the cell and the cell membrane expelling any additional energy. You may be wondering, why is it that passive transport can take place without the cell expending any additional energy? And it all comes back down to the amount of molecules on either side of the cell membrane. When a cell is in an environment that has a different concentration of molecule than inside of the cell, a concentration gradient has formed. That means that if I take a look at the amount of carbon dioxide inside of the cell compared to the concentration of carbon dioxide outside of the cell, and there are two different concentrations, I have an uneven amount, and my cell doesn't like that. And it's going to want to move the molecules from the area where they are highly concentrated to an area where they are lower concentrated. If that happens to mean that there's a higher concentration inside of the cell compared to the outside, then the cell membrane will, without any additional energy, allow that highly concentrated molecule to slip through the membrane to the other side without it having to force it out or move it on its own. If you look to the right hand image, you'll notice that the arrow keeps shooting those carbon dioxide molecules to the right. That's because there's a higher concentration on the left-hand side. There's like 3, 6, 9, there's like 10 of them over there versus the 3 or 4 on the right-hand side. So they will move from a high concentration to a low concentration. There are 3 different types of passive transport, depending upon the type of molecule and or the size of molecules being transported into or out of the cell. The first passive transport is called diffusion. Diffusion takes place when the cell membrane is trying to move a small molecule across the membrane. Because it is a form of passive transport, these molecules will move from a higher concentrated area to a lower concentrated area, either into or out of the cell, depending on the concentration. Examples of molecules that freely diffuse across the cell membrane include oxygen and carbon dioxide. If you look at the picture on the left-hand side, the top cell has a high concentration of oxygen outside of it. You will notice that there are arrows pointing to the inside of the cell where there's much less oxygen. The cell membrane will allow that oxygen to diffuse into the cell because it's going to use the oxygen in order to produce energy. Now, as a byproduct of producing that energy during cellular respiration, carbon dioxide is produced. Look at the bottom picture. That bottom picture has a high concentration of carbon dioxide inside of the cell. So the cell will allow the membrane to let the carbon dioxide leave and equal out those concentrations because we're trying to get rid of that waste product. The cell membrane can diffuse multiple things at the same time, depending upon what it needs. So the oxygen and carbon dioxide will move simultaneously, but just in opposite directions, depending upon their concentrations. The second type of passive transport is called osmosis. Osmosis is a specific type of diffusion, the diffusion of water only. Nothing else will use osmosis. No other molecule, no other chemical will use osmosis to go into or out of the cell. It is just water. Your cells are made up of approximately 70% or more water. So water needs to move freely across in order for your cells to stay hydrated and maintain homeostasis. It will still move from a direction of high concentration to low concentration, but the goal for your cell is to get it so that the concentration of water is equal on the inside and outside of the cell. The GIF on the right shows a structure called a U-tube because it looks like the letter U. The left-hand side is brightly colored red because there's a lot of sucrose, which is sugar. It's got a whole lot of red sugar dissolved on that left side. The right side of the U-tube is clear. That's because it contains distilled water. Over the time lapse of this GIF, you will notice that the water level on the left-hand side gets increasingly higher because the water from the distilled right-hand side is moving across the middle, which there's a semi-permeable membrane there, and it flows across that membrane to the higher concentration of sugar because that means there's less water on that side. The direction the water moves during osmosis is dependent upon the environment a cell is placed in. If a cell is placed in an environment that has a higher concentration of solute, something like salt or sugar that's dissolved in the water, then the concentration of water, the environment is considered a hypertonic solution. Due to the fact that the hypertonic solution contains less water concentration than inside of the cell, the water will move from the higher concentration inside to the lower concentration outside, causing the cell to shrink. If you remember the phrase salt sucks, you can remember that water will move in the direction where there's a higher concentration of solute, because that means that that environment will contain a lower concentration of water. If an animal cell is placed in a hypertonic solution, then the cell will shrink, making it look like a raisin. If a plant cell is placed in a hypertonic solution, it can't shrivel up like a raisin because it contains a cell wall. However, you will notice that the cell membrane and the cytoplasm shrink away from the cell wall, leaving gaps, and that's called plasmolysis. If you notice, the name hypertonic on here has the E bolded and dark. That's because if you see hypertonic, you need to think E for exit, and the water will exit the cell when it's placed in a hypertonic solution. When a cell is placed in a hypotonic solution with the letter O, that means that the environment compared to the inside of the cell has a higher concentration of water and a lower concentration of solute. Because there's more water outside in the environment than inside of the cell, then water will flow inside, because with osmosis, water travels from a higher concentration to a lower concentration. This will cause your cell to swell and get bigger. An animal cell will lysis or burst. The cell membrane can only expand so far, much like blowing air into a balloon, until it can no longer expand, and then it bursts. Plant cells, again, have a cell wall, which prevents them from bursting. However, instead of being a rigid and angular cell wall, it becomes bowed, and we call that turgidity. The pressure from the water moving into the cell kind of causing that cell wall to bend slightly. When you hear the term hypotonic and you see the O in the middle there, you need to think O for grow. A cell will grow and get bigger when it's placed in a hypertonic solution. The goal for any cell is to achieve a state of equilibrium between the concentration of molecules inside and outside of the cell. That applies to diffusion, osmosis, and any other type of passive transport. However, when we're talking about the movement of water and osmosis, if a cell is placed into an environment that has an equal concentration of water and solute as it does inside of the cell, that solution is considered isotonic. That cell is now in a state of equilibrium, where it has equal concentrations of everything inside and outside. Now, because water is a liquid, and when you think of what liquids do at their state of matter, the molecules continue to move and bounce around. This could cause water molecules to enter or exit a cell, causing there to be a slight momentary disruption in the equilibrium. And so when a water molecule moves out, another one will take its place, and water freely moves back and forth across the cell membrane while in a state of isotonic equilibrium. The cell will stay the same size. It will not increase or decrease because there is no overall directional flow of water that causes the cell to either shrink or expand. You may be asking yourself, how do I remember the direction the water moves when a cell is placed into a hypertonic, a hypotonic, and an isotonic solution? Well, I've come up with a cute way of helping you remember that. If you draw yourself a cell, a circle in the middle of your paper, and you label E on the left side and O on the right side. I did it because E comes before O alphabetically. If you do that and you remember E equals exit, then when a cell is placed in a hypertonic solution, as it would be on the left side of the cell, then the water will exit. You will represent the overall movement of water by drawing an arrow in the direction that the water will move, in this case, out of the cell. If you were to place a cell in a hypotonic solution, remember, hypo for grow, the cell will increase in size because water is moving into the cell. To represent the flow of water moving into the cell, you will draw an arrow pointing into the cell, and that represents the cell getting bigger. Now, when it comes to an isotonic solution, water still moves. It moves in both directions. So you represent the movement of water in both directions, not by putting an equal sign, not by leaving it blank, but instead either drawing two arrows, one pointing into the cell and one pointing out of the cell, or you can draw yourself a double-headed arrow. Either one is acceptable, but there has to be an arrowhead pointing and indicating which way the water will flow. Now we're going to pause and check our understanding of the movement of water during osmosis. When you are given a problem that gives you a description of the environment that a cell is placed in and a description of the environment inside of a cell, you need to make sure that you are looking at what's being asked to move. In this illustration, we are looking at the movement of water because it's in the section of our notes that has to deal with osmosis. In order to figure out which direction water will flow, and before we can draw our arrows, we have to first complete the blanks in our problem. And if you look inside of the cell, we tell you that there's 40% solute dissolved in the water. In order for you to figure out how much water there is, you have to do a little bit of simple math. Subtract 40 from 100, and what do you get? You get 60. So you write 60 on that line. Now you do the same thing for the environment the cell is placed in. I'm telling you that there's 50% water in that environment. You do your little bit of math, subtract 50 from 100, and what do you get? You get 50. So you write that on the line. Now that you've figured out your concentrations, both inside and outside of the cell, you can compare the two environments. Ask yourself, where's there a higher concentration of water? Where's the largest percent of water? Is it inside of the cell or is it outside of the cell? Well, 60 is a larger number than 50, so that means there's more water inside of the cell than outside. So, because osmosis is a form of passive transport, water will move from a higher concentration to a lower concentration, which means that the water will move out of the cell. Remember, we draw that using an arrow pointing out of the cell. Part 2, what type of transport is this representing? Well, because we're studying the transport of water, we are looking at osmosis. Here we go. Very last type of passive transport. It's called facilitated diffusion. If you know what facilitated means, you know that it means to help or to assist. Sometimes the cell is going to need to move a large molecule into or out of the cell that cannot fit through smaller pores in the membrane. So to help the molecules move across the membrane, our cell enlists the assistance of proteins. These proteins are called channel proteins, the same ones that we labeled yesterday in our diagram, the ones that look like the little olive cut in half. A channel protein, remember, is like a bridge or a tunnel. It does not move. It is constantly there, allowing you to get from one side of the land to the other side of the land, and it does not change. That's what happens here. Large molecules such as glucose need to move into the cell because it needs to be used as a form of energy in the cell. But it can't fit in between the spaces in the cell membrane, in between the phospholipids and other molecules, so it needs help getting across. When there's a lot of glucose outside of the cell, that concentration gradient makes the glucose want to enter the cell. So the glucose will move its way around the cell membrane until it locates the nearest channel protein, and then it will just slide on through into the cell to where there's a lower concentration of glucose. It is still passive transport because the glucose moves from a higher concentration to a lower concentration, and it's passive because that protein isn't using any energy to facilitate the movement of those molecules. Now that we've completed our notes on passive transport, go ahead and answer the following questions regarding your understanding of passive transport.